Hey, good morning. Questions, actually, video is dealing with the FRQ that I gave you guys last week. Just want to kind of break this down. I didn't do it the week before, and I apologize for that, but I want to get back into that. The question we looked at last week in dealing with hereditary information, if you kind of go through again, the tables, I mean, this is kind of a question I would maybe expect, not this specifically, but just you're getting graphs, getting charts, and you need to be able to analyze what's happening on there. So take a look at table one. It says the data shows the growth of a haploid yeast on media that differ in amino acid content. Plus sign indicates that the yeast strains grow. Minus sign indicates that the strains do not grow. So take a look at the strains itself, the wild type. Notice that it grows on all of these treatments. The mutant one, it basically grows on treatment one. Treatment four does not grow on treatment two and treatment three. And then mutant two again grows on treatment one and three, but does not grow on two and four. So something is missing that is not allowing those individuals to grow in those certain parts. Again, that's kind of what we're taking a look at. Ultimately, we're trying to figure out what is going on between mutant one and mutant two in those treatments to figure out why it's not growing in those certain areas. So if you go to the question, it says a yeast is a single-celled organism. Amino acid synthesis in yeast cells occurs through metabolic pathways, and enzymes in the synthesis pathways are encoded by different genes. The synthesis of a particular amino acid can be prevented by mutation of a gene encoding an enzyme in the required pathway. The researcher conducted an experiment to determine the ability of yeast to grow on media that differ in amino acid content. Yeast can grow as both haploid and diploid cells. The researcher tested two different haploid yeast strains, each of which has a single recessive mutation and a haploid wild type strain. The resulting data are shown in table one. So again, if we take a look at treatment one, no amino acids are present, which basically tells us that mutant one and mutant two cannot, in this case, do not have the ability to produce what they need in order to survive in treatment two. If we take a look at treatment three, looks like all the amino acids are there except methionine, so therefore mutant two is fine, right? It has the ability to produce methionine, right, where mutant one does not, and so therefore it has the inability to grow during that in that treatment. If we take a look at treatment four, all amino acids are present except for leucine. Mutant one has the ability to produce leucine. It can grow. Mutant two cannot produce leucine, therefore, on its own, therefore it does not grow. So if we take a look at A, identify, again, breaking these down into the A, B's, and C's, answering all the bold terms like we've talked about with the past FRQs. A, identify the role of treatment one in the experiment. Well, I take a look at treatment one, we're looking at a positive control in this case. We're looking at a control to where all the amino acids are present. We're basically providing them with a basis that we have given you all the amino acids. Here's what we should expect. So positive control as far as A goes. B, provide reason to explain how mutant one can grow on treatment one medium, right, but cannot grow on treatment three. So again, if we take a look at mutant one, can grow on one, cannot grow on three. Here it has all the amino acids in order for it to survive or to live and to grow. If we take a look at treatment three, there's no methionine, right? Well, if there's no methionine and it can't grow, it's basically telling us that mutant one, based on what we've learned up in this paragraph up here, is that it cannot produce that specific amino acid. So if it can't produce that specific amino acid, it can't survive on treatment three. So again, provide the reasoning. Reasoning would be, again, it does not grow on treatment three because it cannot produce its own methionine, and methionine is not provided in treatment three. So be again, short and sweet, how you can answer B. C is a little bit different. I don't necessarily agree with the answer that some of you have maybe been seeing, like with these FRQs. I really think that there is two possible answers, and the one that like the College Board provides, I don't really feel is right. And I'll explain my choice here, explain my reasoning. So if we take a look at C, is yeast mate by fusing two haploid cells to make a diploid cell. In a second experiment, the researcher mates mutant one and mutant two haploid strains to produce diploid cells. Using the table provided, predict whether the diploid cell will grow on each of the four media. Use a plus sign to indicate growth and minus. So there's two different ways we can look at this. One, if we look at mutant one, mutant two, and they're able to produce a diploid organism, what you're going to see is you're going to find that both organisms or that new organism is going to be heterozygous. So we can look at this in one way. We can look at this as it will have everything from mutant 2 and it'll have everything from mutant 1, which means that it's going to basically be like this wild type over here, which would mean that it can grow virtually on any of these treatments, because mutant 2 will give mutant 1 what it needs, mutant 1 will give mutant 2 what it needs in that new heterozygous individual. So it's going to have a combination of both of those. So therefore it can ultimately grow on each of these 4. So if you're predicting, that would be your one prediction. It's going to be a plus, 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 plus all the way down. And all you'd have to do is just create a little table over here on the side, right? And then you, excuse me, I'll use this. I was using a pen, which you guys obviously can't see. And then you just put in a plus, 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 and a plus over there. I also feel like this could be a plus, a minus, a plus, and a plus. And the reason why you start looking at this is because if you see mutant 1 and mutant 2, well, both of them cannot grow in treatment 2. And so if both of them cannot grow in treatment 2, well, how is it possible for, again, for that new individual to grow in the treatment 2? Because again, it's getting both mutant 1 and mutant 2. So I think, to me, this is the right answer. It's not the answer that they give, but I think this is the right one, because again, you're supposed to be developing a process here and where you can problem solve and be able to analyze these graphs and charts and stuff. And again, to me, that is just simply the right answer, that if you cannot grow, if the mutant 1 cannot grow on 2 and cannot grow on, mutant 1 and mutant 2 cannot grow on treatment 2, then how can that new individual grow there as well? So I personally would put down a plus, a minus, a plus, and then a plus to answer that question. Again, any other questions on this, please ask. I've had a couple of emails on this one. I've went through it and answered individually for you guys. Again, feel free to throw anything at me that you guys have questions or concerns on.